Special guest today is Dr. Marc Weissburg from Georgia Tech. So Marc did his undergraduate at Berkeley, and then he did a PhD at Stony Brook with Jeff Levington. So any of you that have taken a marine biology class probably recognize the name of Jeff Levington, because I think he's written, how many editions of that book?
7, 8.
So after his PhD at Stony Brook, Marc actually came here and postdoced with Dick Zimmer before postdocing a second time at Georgia State with Chuck Derby, and then he wound up in a professorship at Georgia Tech. You're an endowed professor now.
I am an endowed professor now, yes.
Yeah, so that's really big. But Marc's greatest academic achievement easily is the quality of students that he's trained. There are so few of us. Anyway, please help me welcome Marc.
Okay, thank you very much, Lee. One of the quality students right here. I can't think of a better place for Lee to be. He hit the ground running after he left my lab and never looked back, and ended up here as someone who was here at what I think was the very start of this really long and dramatic upward trajectory that you guys are still on. Seeing a student of mine come to a place like this and have the ability to sort of facilitate the science and things like that going on is really gratifying to me for all kinds of reasons. And coming back here, I haven't actually been back here for some number of years. It's really hard to recognize the place. And that's both a physical change as well as a change in the attitude and the activities of people that are here. You've got a great group of young scientists that are just killing it. And there's all kinds of good stuff going on here. And what I've seen has been really, really impressive. And I don't see any reason why it shouldn't continue. So let me talk to you today about this subject here, which is sort of a jargony mouthful. Predator risk cue properties and environmental stress affect the importance of non-consumptive effects in salt marsh communities. Let me unpack that for you just before I get onto it. What we're really talking about is the impact that top-level predators have on ecosystems and interactions as a result of the ability of these top-level predators to control intermediate consumers that exploit the basal resource, right? And that still may be a little bit thick, but we can unpack it a little bit more. This is the kind of situation that I'm talking about here. We have a top-level predator, in this case a spider in a terrestrial system. That spider consumes an intermediate predator, or in this case a consumer, right, an herbivore. And that herbivore itself puts predation pressure on the basal species, in this case grass. So we all know, for instance, that in this kind of system, this top-level predator can eat this intermediate predator, and which consumes the grass. And so there's an indirect and positive effect from this organism here to this organism here that works through this particular link, right? This can happen in two different ways, this benefit of this top-level predator. The most sort of straightforward case is where that top-level predator consumes the grasshoppers, right? Less grasshoppers to eat the grass, right? So the grass benefits. But there's another way that this can be controlled, and that's because this top-level predator can scare the bejesus out of this intermediate species, and that behavioral change can then propagate down to that basal resource, in this case the grass. So these spiders essentially scare grasshoppers, change their pattern of habitat choice, right, away from these sorts of grasses to areas like that. And so this top-level predator, by affecting the behavior of this intermediate prey species, can benefit some species in the community, or can indirectly harm some species in the community, in this case, this flowering species right here. So we call that cascade that works through behavioral changes non-consumptive effects, right? Because it's a behavioral change in this intermediate species that is causing the downstream effect and not the removal of that intermediate species. So I'm concerned about this process in salt marshes. And so those are the indirect effects I talked about. And I'm concerned about this process generally, right? How do we understand the impact of these non-consumptive effects, which appear to be very important? And in order to do that, we have to think a little bit, more than a little bit really, about what information predators and prey can obtain from each other. So what's the sensory aspect, the sensory ecology of these kinds of things, and how does that influence their strength and intensity and distribution, right? So if we think about it, one of the key elements is what is the ability of these intermediate prey species to get information about their predator? What distance is an appropriate distance? Over how far can these prey species detect their predators and respond to them, right? And so I call that reactive range or perceptive range or sometimes predator sphere of influence. They all mean the same thing, right? Over what distance is it possible for the prey species to get information about predator presence? And that turns out to be a really important part of this because it determines the magnitude of these non-consumptive effects. So if, for instance, we have a situation like here where these mud crab species, which we'll hear a lot about in a second, can sense this blue crab predator from a long way away, right? That blue crab predator sphere of influence is very large. Then a large number of these mud crab species are going to be affected, and that's going to make for rather strong non-consumptive effects, right? The behavioral changes are going to be operating on a whole number of mud crabs, and so we'll see a strong non-consumptive effect. On the other hand, if it turns out that this sphere of influence is small, the perceptive or reactive range is small, then only a few mud crabs are going to be affected, and so the behavioral changes are going to be confined to a very small number of individuals, and so these non-consumptive effects are not going to be great. So that, when we first started thinking about this system, that suggested to us that if we understand in the natural environment what this perceptive range is or sphere of influence is, we can then get some insights into how important these non-consumptive effects are going to be in this system. Right? So what is that sphere of fear, that range over which blue crabs can be detected by mud crabs, and how does it influence the strength of this behavioral cascade that we've talked about that goes from here to here? So in order to do that, we do what any good ecologist does. We go out into the field and we ask the animals to tell us what this sphere of influence or reactive range is going to be. And so we set up experiments. A lot of the experiments look essentially like this. Whoops. That's right, I don't have a pointer. We use natural oyster reefs that we have, or refuges, clusters of oysters in which mud crabs can hide, because that's what they like to do. We put oyster spat within those refuge areas, right, so that the mud crabs can forage on them. We place additional spat in these artificial clusters around the periphery that the mud crabs have to go out and find, which is a risky behavior if there's a predator around. And we add predators to this so that the mud crabs are exposed to the risk cues, right, that come from these predators, right? And these are chemical cues from these predators, and we'll sort of talk more about that as it goes on, right? But these blue crabs are emitting scent, and it's the scent of the blue crab, that sphere of fear, that the mud crabs are reacting to, right? So if we do this experiment in the field, and we alter the distance of these blue crabs to these foraging areas, then we can back out how far those blue crabs can be from these foraging mud crabs and still affect their behavior. Obviously, we do this in the field. So this is a typical field setup where you see these field cages right here. They contain these caged blue crabs that emit chemical cues. They contain these foraging areas that are right here. And these distances can be altered. In the other panel, you see how we're adding oysters to these clusters and then placing them in these foraging areas that the mud crabs have access to. Like I say, we do all this in the field. Sometimes we take refuge away from the mud. It's obviously, really nice, great work. All of you have been out there doing it, and it's a whole lot of fun, and everybody has a really great time doing it. Sometimes. This is the result of one of these experiments. And so what you're looking at on the x-axis here is the distance of those blue crabs from these foraging areas in which the mud crabs are allowed to operate. And on the y-axis here, we are looking at the number of oysters that are consumed, right, as a function of that predator spacing. The dots shown over here are the no predator controls, so we can evaluate the effectiveness of these mud crabs in removing oysters in the absence of any predation. Right? And we have both the refuge area in those large clusters and the other areas in which those oysters are found and the mud crabs can exploit. Right? We separate them out for good reasons, but it's probably not something that we need to think about too much. You can get the whole story by simply looking at this particular line and this control here, which is the total number of oysters that are consumed in the experiment. Right? And so what you can see here is that at some distance, the mud crabs are no longer being affected by the presence of blue crabs. So here, in the absence of blue crabs, we get this level of consumption. About 23, 24, 25 of those oysters that we put in there are consumed by those mud crabs in the absence of any interference by the predator. Right? If the predators are close, right, then the mud crabs are sensing those predators and their foraging is suppressed. Right? It's actually suppressed more outside of the refuge than in the refuge if you look at this figure carefully. And that suppression of behavior extends all the way up to about this distance here. This is the greatest distance at which we see that total consumption being different from the no predator control. So we take that as an estimate of the range over which these mud crabs are perceiving or at least reacting to those predators, right? So that's our reactive distance in the field under these conditions, about 1 meter. Any distance greater than that, the mud crabs don't care about the predator presence. Any distance less than that, the mud crab foraging behavior is suppressed by the presence of chemical cues that come from those blue crabs. It turns out that this reactive range is affected by the biomass, the amount of mud crabs that these predators have consumed. So one of the things that we did in this experiment was to feed blue crabs a lot or feed blue crabs a little before we put them out into the field, and that manipulates the amount of scent that's coming from those blue crabs. Right? When we feed the blue crabs less, they're less scary. Right? This suggests that the dietary metabolites that come from predators digesting their prey are what's responsible for those chemical signals. And that becomes important later on. Right? It also says that there are feedbacks between the amount of material that a predator consumes and its scariness, as you might expect, although it kind of works in sort of the opposite direction. If you're really, really hungry, you can't be detected very easily, and so you then become very vulnerable as a prey species, right? If the blue crab is really well fed, then they're really detectable, and you can run away from them, right? And so there's this sort of natural feedback loop that regulates the intensity of this response. The less you eat, the less scary you are, the more you can find prey, the more scary you become, and so the effect sort of oscillates, right? Or at least one presumes, right? It's pretty clear here, right, that the sphere of fear for a blue crab that has not eaten a whole lot is, about 0.5 meter, right? Quite a bit less than what you see when the predators are well fed and emitting lots of metabolites. So, what I've shown you, right, is that predator cue can be used by the prey to determine the amount of threat that is out there in the environment. What modulates this, right, aside from, let's say, the predator diet, right? We're out there in a salt marsh. There's lots of stuff going on. There's lots of flow. There are effects there that are important, potentially, right? And so we can talk about two different things. We can talk about sensory stress, which is actually a phrase that Lee coined and has been useful for us for many years after that. Fortunately, he didn't trademark it or patent it or anything like that, so I can pretend that I made it up, which I really didn't, right? So there's sensory stress in that some levels of fluid motion disrupt chemical plumes and make it harder for an organism to sense them, right? They're not stressful in a traditional sense. They don't alter the ability of the organisms to move or do anything else, but they do affect their ability to gather information. That's a sensory stressor in the same way that, when it gets dark outside, you can't see, right? That's a sensory stressor, right? When that fluid flow becomes extreme, then the ability of the animals to, let's say, locomote or even forage is affected, and that's what we would call sort of a physical stress, right? That's the kind of thing that happens on rocky or tidal coasts where wave bashing, makes it hard for a predator to move, right? And so the question is, are these effects important? Right? Is this physical forcing important? And does it exert its effect by altering the ability of the animal to get information or to act on that information? Right? Sensory versus physical stress. So in order to start to think about that, we can look about how this reactive range changes across a flow gradient. Right? If flow is a sensory stressor, then it should change that reactive range. Right? And again, there are good reasons to think so because fluids disrupt chemical plumes. Right? And so what that means is that has the potential to change whether the predator is exerting its effect through consuming mud crabs or through scaring them. So we've been working out in Skidaway. This is Wausau Sound. It's located essentially right coastal to Savannah, Georgia. Right, that's the field station that Georgia Tech has had access to. That's the site where Lee cut his teeth as a beginning scientist, as well as other body parts. We've been working out there for 15 years, and we know this environment very well. And in particular, we've monitored the flows very carefully in a number of places in this marsh, and we can identify places where the amount of fluid flow is very, very different and has the potential to sort of act either as a physical or sensory stressor. So, for instance, in this particular site here, which is actually adjacent to the Skidaway Institute of Oceanography Marine Lab, we see higher levels of fluid flow, higher current speeds, higher turbulence intensities, things that may actually be physically preventing organisms from moving or foraging, whereas at this particular site here, right, which is out on the Wilmington River that's heading out into the Atlantic Ocean, the fluid conditions are a little bit more benign, and there are things that could potentially affect the sensing, the ability of mud crabs to detect the blue crab prey, but probably aren't sufficiently high in order to suppress the ability of mud crabs to forage for oysters. Okay? And so we use this variation in flow, which is also produced by variation in tidal amplitude, to do some natural experiments, right? At any given site, the greater the tidal, the amplitude, the higher the current speeds. And we know this from lots and lots and lots of monitoring. And we can essentially now, as a result of all the data that we've collected, go out into an area. If we know what the tidal height is, we can make a relatively robust prediction about what the fluid flow is going to be like. Right? So we have a good way to correlate in a given instant of time what our physical conditions are. Right? And we use that to our advantage when we do these experiments. So again, now we're looking across this flow gradient as a result of tidal amplitude and site variation, and we ask, using the same techniques that we've used before, what the reactive range of mud crab prey are to these blue crab predators. Right? And so again, here's one of my erstwhile field assistants. You know, never bet against a person who looks small of stature. My best field assistants have typically been people who are not even as big as the cage, but they're hauling 3 cages out there in the time that it takes me to walk out there. I think maybe they're just lighter and they don't get stuck in the mud or something like that. This is Bailey Lynch. She was a really great field assistant, right? And cheerful, as you can see, despite the fact that she's put out 20 of these cages. And, you know, I'll point out that the distance from one end to the other is probably about 1/4 to 1/3 of a mile, and you're hauling out all kinds of gear, right? So if you really like field work, you know, come look me up, because I could definitely put you to work. So here's a somewhat complicated figure, but what we're looking at are the fluid flow parameters that correlate best to us with sensory versus physical stress, and what we know about the perceptive range in those environments, and what we know about the ensuing oyster survival, right? And so again, remember the paradigm is that the blue crabs are present. They're allowed to produce chemical cues that can modulate the mud crab behavior in these conditions where we know what the perceptive range is, and then we're looking at that perceptive range and trying to understand relative to those physical parameters what's causing this change in oyster survival. And so one of the first things you notice is, again, these conditions correspond to different perceptive ranges, right? So for instance, in these two conditions here, right, as a result of this turbulence level, those chemical signals get all disrupted very easily, and so even if you're really close to a blue crab, you're not able to sense its odor, right? Actually, the closest distance that we're able to back out is 25 centimeters, right? And so we know that odor signal does not even propagate in any effective way a quarter of a meter away from that blue crab, right? So that perceptive range is essentially zero, right? On the other end of the scale, right, so you see here when the turbulent kinetic energies, a measure of turbulence is less, right, that perceptive range is large, right, up to 2 meters, right, in our most sort of benign condition here, right? And you can see that there's a strong influence of that perceptive range, in this case labeled as reactive range, on the oyster survival. So here, the reactive range is really large, right? And so oyster survival is really high because the mud crabs can readily detect the presence of that blue crab predator and their foraging is suppressed, right? Which is benefiting the oysters. Here, there is high oyster survival, but it doesn't have anything to do with the presence of the blue crabs because the perceptive range is zero, right? So what's actually affecting the situation here is not that the mud crabs no longer sense the blue crab predator, or rather, the mud crabs sense the blue crab predator and don't forage. It's that the mud crabs physically cannot forage at all given the levels of fluid flow out there, right? So here, at this level of physical stress, the presence of the blue crab is irrelevant because the mud crab just can't physically operate. So we flipped a switch, right? The predator in one case has an effect to the propagation of information. In a more physically challenging environment, the predator has no effect because the mud crab can't do anything. In the middle, here, we see that there is some impact of mud crab perception, right? But it's not great, right? The oyster survival is low, right? Because the mud crabs are foraging because they no longer sense the presence of that blue crab predator. So in this particular fluid regime, where we have some turbulence, but not as strong a velocity, right? We see a sensory stress, but no physical stress. And so depending on these particular conditions, we're able to flip the switch essentially in how well the predators are able to control that mud crab foraging and what the downstream benefit is for the oysters, simply by altering either the information flow in this environment or the ability of that mud crab to actually react, right, and forage in these situations, right? So the physical environment is controlling both the strength and potentially the way in which predators are affecting these basal resources by constraining or not constraining mud crab behavior. So in other words, we've changed the magnitude of these non-consumptive effects, right? One of the questions that we initially had when we started out this work is, are these non-consumptive effects really important, and when are they important, and why are they important, right? And it turns out that when we first started this work, it was a little bit discouraging because it seemed like everybody had answered those questions, right? That there was this universal feeling, as you see here, that these non-consumptive effects are really important, right? And of course, the idea is, as you see here, right? Because these behavioral changes, because many animals out there can potentially gather information about the predator presence, we expect these non-consumptive effects to be strong because many organisms are simultaneously affected, right? And that signal should be stronger than the signal of direct predation because an animal can only eat one animal at a time. Right? Now, those of you who have been thinking along with me are probably now thinking, but wait a second, but you've shown that reactive range can be really, really different, and so there are clearly some situations in which the mud crab prey, that intermediate prey species, can't sense the predator. So why is it that people think these non-consumptive effects are always important because lots of organisms are affected, right? Because you've just shown the opposite. That's exactly right. I have shown the opposite. And one of the things that we realized, and this is a result of a collaboration between Lee and myself and another former graduate student, is that the way that we've obtained information about the importance of these non-consumptive effects has not really translated into really strong predictability in the field, right? And that's because of the way that people do experiments, right? These are hard experiments to do, right? I mean, you're out there covered with mud, right? And unless you're a really cheerful person like Lynn Daly, you don't really want to do that, right? You just want to do an easy experiment in a mesocosm, right? And you'll say, yeah, mesocosms are easy, right? And a lot of the experiments that people have done, particularly in aquatic habitats, and most of these experiments, most of the information that we know about NCEs comes from experiments in aquatic habitats, either stream or lake or in this case, our case, the estuarine environment. Most of those experiments are done in physically very unrealistic conditions, right? No fluid flow, right? Or unquantified fluid flow. And so unintentionally what we've done in the hundreds of studies of non-consumptive effects has been to bias these results toward conditions where it's really easy for those prey species to sense their predators, right? Whereas if I go out and do that in the field, that's not going to be the case in lots of situations, right? And so it turns out we don't really know as much about the importance of these non-consumptive effects in nature because we've biased our experiments to obtaining a particular answer that may not be applicable, right? And yes, that observation has not made Lee a really popular guy. Actually, it's not made me a really popular guy. Lee's caught a little bit of flack for it. But it's true, right? I mean, if you want to explain the world, we can't explain the world through doing experiments in mesocosms. They're really useful for some things, right? But if you want to explain nature, you better go out in nature and do it, right? And, don't complain that it's hard, because if it was easy, it'd have been done by now, right? And so that's why we do things in the real world, right? And so now, knowing that perceptive range has been changing, right, and seeing these different kinds of effects, we thought it was important to really compare, right, the role of predators that exert an influence by directly consuming the prey, these consumptive effects, versus the effect that's propagated by predators changing behavior of those prey species. So which one of these effects is more important, right? And under what? And that's really the trivial question, right? The important question is, under what conditions is one of these things going to be more important than the other? And can things be driven toward one of those other effects depending on the physical environment? Because understanding that has lots of important consequences, right? So we do the same kind of experiment that we've done before, right? But this time we do it in a slightly different way that allows us to back out the strength of this consumptive cascade versus this non-consumptive cascade, right? And our sort of hypothesis was that this is going to depend on the level of sensory stress, right, in a way that's related to the perceptive range, right, and the physical stress. Right? And again, we utilize the same habitats that we've used before because we have a good understanding of what the fluid flow regimes are. And the experiment is a little bit more complicated now, right? And so now in each of these different sorts of fluid regimes, we set up the following set of treatments that you see here, right? And those are designed to explicitly separate the role of direct consumption versus the indirect effect as a result of changing mudcrab prey behavior, right? And the rationale behind this experimental design is a little bit too cumbersome to go into, but this is really the way that people advocate trying to directly understand the effect of consumption versus non-consumption in these kinds of experiments, right? And so those are the four treatments we use, right? A control where there's no predator, a treatment where we manipulate the risk cue, which we call the non-lethal blue crab. We have chemical scent, but the blue crabs can't consume the prey. This is essentially all the experiments that we've been doing previous to this, right? We add that blue crab predator to the mix, which is producing both a consumptive and a non-consumptive effect, right? It's eating things and scaring things, right? And then the last treatment is this sort of fictive predation thing where we manually remove the mud crabs, right, on a schedule that approximates blue crab predation, but that treatment has no signal to it, right? We just go in there and grab the mud crabs out, right? And so that is an effect that only is propagating this consumptive effect, right? What the blue crab would do by consuming prey species, mud crab prey at a certain rate, okay? And then we can compare these all statistically which, again, I won't bend your mind about because it makes me crazy. Okay, so again, we find that these things are related to the fluid physical environment. So oyster survival is increased by these non-consumptive effects, but only at levels of fluid motion where there's no sensory stress. Right? And so what you can see here, right, is that the oyster survival is enhanced when we scare those mud crabs, right? When we only have the Q present. There's not much of an effect of the ability of the blue crab predators to consume those mud crabs because the mud crabs are hiding in that oyster shell matrix and can't be eaten. But if they're hiding, they're not eating the oysters, right? And so that's where that effect comes from, right? When we add the predator here, it's essentially no different than if we just put the predator scent in there, right? Which tells us, again, that what's happening here is a result of fear and not of blue crabs consuming those mud crab prey. Now, right, and so that cascade, right, is happening indirectly as a result of behavior, those blue arrows, and not as a result of consumption. Right? Now, again, at these levels where there's some fluid motion, right, enough to disrupt the sensing of these mud crabs, we see, in fact, that most of the effects are exerted by the blue crab consuming those mud crabs, right, as you would expect. They're not being scared, they're being eaten, right? So with this change in the physical environment, we flip the way that that blue crab predator is exerting its influence on the prey species. Right? So that red link, right, is operative, and there's no fear effect. Finally, when we amp up the fluid motion enough, there's no effect at all because the mud crabs can't even forage. Right? So the blue crab presence has no effect on the system at all, right, as you see here. Okay? Right? And again, if you go back here, we see that the fear effect does nothing, the consumptive stuff does something, and that's approximately equal to the natural predator. Right? If that's not enough, we can compute effect sizes, right? And so some of you might realize that there's a way to take the log of the ratio of responses in various treatments and obtain a statistical quantity, which is also called which is sometimes called the effect size or the log effect size, and that allows us to compare the explanatory power of different treatments in a statistically relevant effect. And so what you can see here is basically what I've told you, right? A positive effect is blue crabs exerting a beneficial effect on the oysters, right? And so here in this very benign condition here, we have a positive effect on oysters because the blue crabs are scaring the mud crab prey. Right? When we have a slightly larger level of fluid motion that's enough to disrupt the mud crabs, the effect is positive for the consumption part of the chain, and over here we have no effect whatsoever. One of these days you might even see this in print, God willing. But then we have, anyway. Okay, so in a visual, right, so here's the visual summary of what I just told you. Okay? Again, I think this is really important because it gives us intuition about how we expect predators to function and when and why, right, which is something that I think we've been missing. And I think this general principle applies sort of uniformly, regardless of environment and regardless of modality. If we think about vision, we can think about gradients in visual sensory stress. Lee has thought about that with respect to turbidity. Right? And this occurs both in terrestrial and in aquatic situations, right, where there are gradients in the ability of organisms to see. Right? If we think about terrestrial situations, odor can be really important. Wolves in Yellowstone, for instance, right? So, you know, what we need to do is be thinking more about how that sensory environment is affecting things and designing our experiments at least so we're aware of what it is that we're asking the organism to do rather than simply assuming that, let's say, what we find convenient, right, which is a mesocosm, is representative of the sensory environment that the organism is experiencing in the real world. If we don't do that, we're going to get the wrong answer. So I'm going to shift gears now because there's another aspect of this chain that's important, and that's the role of the quality of that chemical signal, right? And so the predators release a cue, and that cue is available for the prey to detect, in this case mud crabs, and that results in some judgment of risk, which then modulates the response, right? And so what we're concerned about is what is really going on here, right? What's determining how risky these organisms think the environment is. And that's concerned with characteristics of that predator cue, right? The predator determines that in any number of ways. So what are those links, right? I've shown you one. The amount of biomass that's consumed by the predator strongly affects the assessment of fear, right? When prey species are getting a big whiff of, metabolites from a blue crab, they're really scared. Right? Turns out that there are other kinds of effects that are important here too. Right? So if we look generally at this, and this is from Nancy Shopler's work and Rick Rayla, with respect to this dragonfly tadpole system, dragonfly larvae are voracious predators of all kinds of things, and there's a really, really well-developed response of tadpoles to smell and see and hear, if you will, the presence of dragonfly and other kinds of things that are willing to eat them. These tadpoles, like many other organisms, have potentially two sources of information available to them. One of them is, my buddy's been injured. That smells bad. I'm going to run away from that, right? The other is, there's some scent in the water that feels a little bad for me. I'm going to run away from that, right? And these are presumably distinct sources of information. And they're somewhat uncoupled from each other, right? I mean, you can be injured as a prey species, not as a result of predation, and you can be a predator that's leaking metabolites that don't have to do with a particular prey organism. Right? So both of these things can be modulated, and clearly, if that dragonfly nymph is eating that tadpole, both of those cues are going to be combined. There'll be both a predator signal as well as that injured metabolite signal. Not going to talk about the injured metabolite signal because we don't know much about that yet, but I will talk about this predator cue. Right? If a predator eats different things, and we all have this experience with, let's say, asparagus, right? Your metabolite smells different, right? Right? So there's clearly an influence of the diet here, and that can be a potential cue to the presence of a predator that's specifically consuming your buddies, right? And so if a predator's out there that's not consuming your friends, maybe you don't pay attention to that. It's not as much of a mortal threat. But if, hey, Bob just got eaten, man, I better pay attention, right? And that's kind of the logic of thinking about predator odor as a result of diet differences. Right? I'd like to tell you we know a whole lot about these cues, but we don't. We know virtually nothing about them, right? So there's this whole world of chemical ecology out here that is very opaque to us. Right? There are 16, this is, I looked at this data about 2 years ago. There's 16,000 chemicals, metabolites, that have been identified from marine organisms. That's a lot. Right? There's a whole cottage industry of doing this. Some fraction of them, about 1/8 of them, are feeding deterrents. Right? That is, stuff in the flesh of organisms that is yucky, and that when a predator takes a bite out of it, they're like, no, I don't think so. Let me find something else, right? That's right. So we know something about what deters predation, but not in the way that I'm thinking about, right? The effects that I'm thinking about are a fear effect, right? This isn't a fear effect. This is the effect, right? What do you call it? The ecology of Yiddish. Good Yiddish word there. Okay. There are 9 metabolites that actually are fear-inducing. Right? So we've gone from 16,000 identified things to 10, right? A fraction of a percent that we know the identity of that produce fear. Only 2 of these are waterborne. 0.001%, right, of the chemicals that we know about are waterborne fear deterrents. That's a pretty small number. Why don't we know about these things? Is a question that I'll get to in a moment. But I want to point out that the feeding deterrents that we know about are all really specialized molecules. That is, molecules that are produced by the prey species that are specifically designed to dissuade predators from eating them. They're all what we would call secondary metabolites. They have no other purpose that we know of except to deter predation. And they're produced by the prey species itself. right? That distinction is really quite important, and we'll keep it in mind. And some of the molecules you see here, really complex, difficult to synthesize, very specific, large-ish molecules. And they come from the organisms that you see there, mostly algae, as well as a few fleshy things. The fear-inducing molecules that we know of are also specialized to some extent. Only 2 of them are waterborne, right? These are the molecules that are produced by predators, right? That cause the prey species to be aware of that predatory activity and run away, right? And again, a lot of these are pretty specialized. The 2 that are waterborne, copepod amides and aliphatic sulfates, come from phytoplankton, right? That are concentrating certain nutrients, right? So these seem to be pretty specific cues. And again, their structures are rather unusual. Right? Why don't we know about all the hundreds of other kinds of things out there? Again, there are literally 500 systems in which we see waterborne chemical effects of fear. We know nothing about any of these chemicals, right? And we know nothing about why things are different if you eat flesh of a different type. Right? So I've already told you that what the predator is doing is important, right? Again, we have this indication that the scary molecules are coming from the flesh of the organisms that are being consumed, because that seems to be, it's hard to figure out why you would have this diet effect if that's not the case. Right? So that got us thinking about this a little bit more carefully. Right? We realized that if we could find a situation where predator diet was important in our system, and we could directly obtain the metabolites from the organism, from the blue crab, we could apply some really good chemical techniques that might answer the question that are difficult to apply in other cases. Right? And so it turns out we do actually have both of those conditions in this system, which was quite a breakthrough, right? Really, in this, as you'll see in a moment, right? What you see there is the tip of this forceps is. Can you see this, Lee? Yeah. Okay. Good. That's the nephropore of the blue crab. You can see this. You could see it on the animal, right? We're teaching Lee how to do this so we can work together, and it's very challenging. That's the site of urine release of these organisms. It's right below the eye sockets and around the gill chamber, and so that's where the organism is peeing, right? So we can catheterize that, and we can obtain the urine that contains all those molecules in a highly concentrated form, right? Which is a real advantage for chemistry, right? And it turns out, in fact, that as you see here, if we give the organisms different diets, the mud crabs are scared to a different extent. Right? And so what you see here are dose-response functions for the amount of urine and how that amount of urine is producing feeding suppression in mud crabs. And so what you can see here is that if we give the oyster diet to these blue crabs and harvest their urine, yeah, it's scary. There's something in there that the mud crabs are responding to, because as we add more urine, right, that feeding suppression continues to be more intense, and at some point the mud crabs are just like, I don't think it's worth it. I'm going to stay home and watch TV, right? But the dose-response function for the blue crabs that have consumed the mud crabs is a lot more intense. It's a scarier signal for some reason, right? A given dose produces a much greater response when we fed the blue crabs mud crabs than if we have fed them oysters. Right? And there's 2 possible reasons for this. It could be that the blue crabs are just producing more stuff, right? Or it could be that the chemicals are different. And so we tried to answer that question, because then we understand what the molecules are like. And so what's the nature of this diet effect? Well, that's kind of hard to figure out, because this is the NMR spectra of blue crab pee. There's a lot of stuff in there, right? And so to try to figure out which one of these peaks, and there's several spectra overlaid on top of each other, and they're all very similar, right? They all have peaks in the same location. But there's a lot of different things going on there, right? So how do you figure out which one of these peaks is tagging a molecule that is producing this fear effect? Aha. Well, now I must tell you about metabolomics. Okay? So some of you probably have heard about this. It's a slightly different approach than the way that people historically have gone about trying to figure out what bioactive molecules are. So in the metabolomics approach, what we do is sort of a statistical kind of method where we take a sample, right? And for each sample, we obtain both a chemical profile of that we can treat with multivariate analysis, and an assay that tells us how the organisms are responding to that single sample. And so what we end up with then is this sort of multivariate analysis, right, where for every sample we have, right, we have a set of NMR data that gives us a profile. Each one of these peaks is an element in that profile that we can sort of look at it in a multivariate statistical sense. And we can ask what portions of that spectra explain the difference in the bioassay that we get. Right? And so this sort of variance partitioning approach allows us to say, this feature varies, and that variance of that feature is associated with variance in the bioassay that we see. Right? And so this means we can work with very small amounts of material. Right? We don't chemically degrade, fractionate, do anything that will further reduce the amount of material. Right? And it's a very powerful way as long as you have sufficient variance in order to figure out what's going on, because multivariate analysis without any variation tells you nothing. Right? This is sort of in contrast to the traditional approach where we take a sample where we need a huge volume, we chemically treat that sample, pull out a certain fraction, test it, fractionate it some more, test it, fractionate it some more, test it, fractionate it some more, test it, right? So at the end of the day, we might have this much sample left, which then we actually have to purify and identify, and we need a whole lot of stuff at the beginning. And that whole lot of stuff can't have any water in it because chemists hate water, right? It messes up the analysis, right? And so the reason that people haven't identified these compounds before is because they haven't applied a metabolomics approach, because they don't have any variation, or they don't know if there's any variation, and they can't get the pure compound, right? So if you think back on papers you've read about this, what do people do? I mean, I've done this, right? You stick your predator in a bucket, right? And then you take the effluent, and then you apply it to the organisms, and you see what the organisms do. But that doesn't work because you've got so little stuff and so much water, right? And so we were able to use this metabolomics approach to figure out what's going on, right? So this is sort of the beginning of that process. And so what you can see here is blue crab, the urine of blue crabs that have been fed two different diets. This is the oyster diet, this is the mud crab diet, and we're doing essentially a PCA on the NMR spectra asking if they're different. And they are, right? There are molecules that are everything has the same set of molecules, right? So they're not qualitatively different, right? There's no unique molecule here that identifies predator urine from a mud crab diet as being mud crabs, right? There's no mud crab molecule, if you will, right? There are only a whole bunch of molecules that these two things have in common, some of which are more abundant and some of which are less abundant. And that's what you're getting there from that NMR trace. But they're clearly distinguishable. And again, you can see a series of traces that are overlaid on top of each other, right? And you can see that the two traces themselves, the orange trace, which is the mud crab, and the yellow trace, which is the oysters, have the same peaks, but smaller or larger, okay? Quantitatively different. These are the molecules that we were able to identify as being quantitatively different. So these are the candidates that can explain why one urine source is scarier than the other. And the ones that are upregulated in the mud crab diet are the ones with the red arrows, and the ones that are upregulated in the oyster diet are the ones that are in yellow arrows. Now, if you look at this list, what you can clearly see is these are all really common molecules. Right? There aren't all these crazy specialized molecules that you get when you look at the flesh of red or green algae, right? And if you think about it, this makes perfect sense, right? It's not the predator doesn't want to produce these signals. The predator doesn't want to scare the prey. It's the prey that's trying to listen in, if you will, on that predator, right? And so how does the prey best do that? Well, the prey best does that by finding molecules that predator has to excrete as a result of what happens in the digestive process. And so that's where these molecules are coming from, right? Which means that they're liable to be ubiquitous and universal, right? Because on a fundamental level, most organisms, unless we start thinking about some really crazy ones, have pretty similar metabolic processes, right? And the composition of prey flesh, while it varies to some extent, does not vary dramatically, right? Again, unless we're talking about some very specific sorts of organisms that might sequester a certain molecule, right? But certainly that's not going on in oysters and mud crabs, right? So these candidate molecules are exactly the ones that you would want as a prey species if you want to eavesdrop on predator activity. And again, they're ubiquitous, they're universal, we think, because they're coming from these primary metabolite processes, and of course they're benign, right? They're not things that are poisonous or toxic to you. Right, which has lots of advantages if we want to think about, let's say, conservation problems. And I'll wrap that up in a second, right? So what we can do is, again, apply the statistical model to these peaks, right, and say, which one of these peaks correlate best with the bioassay that we've seen as a result of our model? And we ended up with 3 really good candidate molecules where we thought the activity could be explained easily by the concentrations that we were able to find in the urine, right? And so here you see the predictions of the measured urine potency versus the predicted urine potency for homarine, TMA, and trigonylene. So those became the molecules that we decided to test. It's pretty simple. We give these molecules in the same concentrations that we were able to find in blue crab urine, and we see if it produces foraging suppression in mud crabs. If it does, those are the molecules, those are almost certainly the molecules that are responsible. Not that there might be other ones that aren't being detected, but these ones certainly work. And that's what Remy Poulin did, and that's the result, right? And so we were right. Okay, I think I can go all the way to here. We were right for 2 of them, right? So trigonylene and homarine, which are structural isomers of each other, predict very well the response of mud crabs to urine, to whole urine obtained from blue crabs that have eaten mud crabs, and trimethylene does nothing. So now we have molecules that we know are responsible for this ecology of fear effect, and they're things that have a variety of not only important sort of ecological implications, but maybe some important implications if you're thinking about oyster reefs. Right? And so one of the reasons that Lee and I have been talking to each other is that Lee has found a similar kind of thing that's going on with respect to blue crabs and other organisms that exploit oysters. The oysters are responding to some of these signals too. Right? We're not sure which ones, right? But the data that I presented to you today suggests that some of these molecules, and this is data from Lee and Avery, some of these molecules might be the same. And so we're very interested in seeing if we can produce these effects using the molecules that we've identified or identify some of these other candidate molecules and use them for things like oyster conservation and management, right? If we can suppress predator activity, right, then we can enhance the ability of oyster reefs to accrete. If we can make oysters tougher in response to predation, then we might be able to enhance their survival, right, when we put them out in the field. And so in addition to trying to find out what these metabolites are and understand the ecological implications and what the rules of signaling are, we'd like to be able to use this in a management and conservation aspect, right? And this is not uncommon. I mean, people for years and years and years have been using chemicals to disrupt processes in insects, right? So this is something that has shown to be successful and is likely to be successful in this particular situation too, if we can package these molecules and apply them in the right ways, and that's what we're hoping to do. Let me acknowledge quite a few people who have done most of this work, especially my colleagues Remy Poulin and Julia Kubinik, these are the chemists that have been involved in this project. The chemistry here is not for the faint of heart. If you don't have a PhD in chemistry, you're not going to be able to do it by running gels on a colony or something like that, right? And so, these people's expertise were really critical in doing this. Jessica Pruitt was responsible for all the really beautiful work on physical forcing of consumptive and non-consumptive effects. She had some help from an undergraduate student, Jeff Beauvoir, who helped look at some of the perceptual range phenomena. And then over the years, we've been fortunate to have support from a variety of people, both in my lab. Alex Draper was originally a student of Lee's, and I was lucky enough to be able to steal him away from Lee when he decided to move on, as well as people at Skidaway. I mean, all of you guys work in a marine lab, and you know how important the support is from the staff that's out there to help make your work easier. Without people at Skidaway, we would not have been able to accomplish this work. I'd like to thank you for your attention, right? And hope there was something interesting there for you, and I'd be happy to take any questions.